Well, next up on our agenda, we're really excited to have Dr. Dorrian Garrick here, J. Lush Chair of, now Chair of Animal Breeding and Genetics at Iowa State University, and he's going to share with us his wisdom on conventional EPDs and genomically enhanced EPDs, and he is right in the thick of all of that, so it should be a good talk.
Good. Thanks very much, Jackie, and thanks for the opportunity to come and tell you a bit about some of the work we're doing. And I'll just try and sit down and turned on. Understanding conventional and genomic EPDs. It's worth stepping back for a moment and just thinking about the big picture of selection, because EPD is just a tool, and having EPDs doesn't give you improvement. It's using them appropriately that gives you the improvement. So this is just a depiction, very simply, of the problem that you focus on. On this axis here, I've got income. I've said income is the collective consumer satisfaction.
Hey Dorrian, your mic's off. Can you?
My mic's off?
Yeah,
It's got a red light on it. Is that good or bad?
We're not picking you up through the mic system anyway, I don't think. Try going closer with it. Yeah, now it's picking you up.
Yeah, this is Okay, so let's just try and depict the selection problem that you are hopefully trying to deal with. So on this axis here, I've got income, and I've expressed income as the sum of all the consumer satisfaction that comes from the eating experiences that you sell. Because that's what the beef business is about, right? Even if you're selling wieners, it ends up on somebody's plate. Ween limbs, I suppose I should say. I've got to be a little bit careful coming from down under. And on this axis here, I've got the sum of all the costs of production. So all the feed, the replacements, all the other things. So each of these spots might represent the mean of the offspring of a particular sire. So this offspring has a particular, all of the offspring of this bull, they have a particular cost of production and they have a particular income. So it represents a cloud. And what you can see is there's a whole lot of variation in cost and there's a whole lot of variation in income. So that gives us an opportunity to do selection. You'll notice it's a cloud, it's kind of tipped over a bit. It's not a circle, and that's because there's a correlation between these traits. So the genes that are associated with more income tend to be associated with genes that are associated with more cost. So what we're trying to do is move that cloud. And what we know from experience of 100 years or more of selection experiences is that we can move that cloud in any direction you like, except we can't shrink it. Selection is particularly ineffective at reducing the variation, but it's very effective at moving the mean. So we can move it up, we can move it to the left, we can move it to the right. And so if I took the difference between the income and the costs of these points, it was about $490 on that particular set of assumptions there. So how are you going to move that cloud? So one of the things that might appeal to you to move that cloud would be independent culling levels. Now that's not the idea that Hazel, who did his PhD more than 65 years ago, came up with the idea of selection index theory. And he said what you should do is you should rank all of these bulls on what he called their aggregate economic value. Well, nowadays we think of it as their index value. And he said we should measure all these traits on the animals and use them to predict that overall value. And all you would have on your sire summary is the overall value. Unfortunately, to implement it, you needed to know the economic values. And somebody who came through a PhD a little bit later than him, a guy called Henderson, showed that you could actually partition the problem into two pieces. One was to get the EPDs, and that didn't involve any economics. And the second piece was to use the economics to combine the EPDs to get an index value. And he said he didn't know anything about economics, so he'd do the EPD bit and leave it to the economists to come up with the economic value. Unfortunately, they weren't listening. So that's why for the next 30, 40 years, what we got was EPDs and not indexes. And that, unfortunately, was a little bit of a step backwards. And one of the interesting things listening to Bruce's talk is that theory on selection index came out, in the 1940s. And what was the date you said the first index came out? 2004, right? So that shows you the generation of people it took you guys, whoever they are, you guys, to implement this stuff. Now compare that to genomics. When did genomic stuff become available in cattle? 2009. And it was implemented a couple of years ago. So we guys did a hell of a lot better than you. Okay, so one of the things, if you didn't know about indexes, the easy thing to do is independent culling level. So you might say, I want to take all of these bulls who had below average cost of production. So that's everybody that's less than average. But I also want them to have above average income. So that would be the black spots. And if you chose those animals to be the parents of your next generation, their profit averaged 658 instead of the 492, which is the average of the whole lot. So if you use those guys as the parents of the next generation, you'd move the cloud up and to the left. But it would still be the same size the next generation. The variation would be reconstituted. If we were to use Hazel's idea of this aggregate economic value, then we would rank each of these animals on their profit, which would be the difference between income and costs, and those are the black spots representing who you would select, and then the profit of the ones you selected would go up to 677. So selection index is more efficient than independent culling levels if you want to move the selection, and it's most efficient when you use the index values for selection without all of the component traits. So Bruce told you that if you have ERTs and indicator traits, you make better selection decisions if you don't have the indicator traits. I'll go one step further and say if you have your index properly constructed, you do a better job of selection if you don't have the EPDs on all of the ERTs. But that might take another 60 years for you guys. Okay, so we know that the more efficient way to move that cloud, which is what we're trying to do, is with EPDs. Right? EPDs are more effective than phenotype at moving the cloud. Phenotype is associated with real genes through something we call the heritability. And the heritability is the strength of the association between phenotype and genotype. But to move the clouds, genotype we need to be dealing with. So EPD is an old idea. We have a bull here, like this one here. He's got a bunch of offspring, and let's say maybe he's got 100 of them. And those offspring are going to show a lot of variation in performance. And that variation is due to how old the mother of the calf was, how early it was born, and a whole bunch of other things, the sex of the calf. And those things are not genetic things to do with the sire. So we have to remove the noise from that. So that's what these things would be, the adjusted deviations of these animals. And one thing that surprises a lot of people is if you did this with a real bull, there's a huge amount of variation there. And we can show that just in terms of the genetic values, there's 3/4 of the diversity of the whole population in the offspring of a single bull there. So a lot of people have the idea that a good bull has good offspring and a bad bull has bad offspring. And if you ask some commercial people, they've got a bunch of bulls, they think they can tell which calves came from the good bull. They don't recognise how much variation there is here, but there isn't a whole lot. In real life there's a whole lot. But we can average all those, and that gives us kind of a summary of what the performance of all those offspring are like. But that's not the best way to do selection. Because when I hear something, I have to decide how much am I going to trust it. So if I hear Willie tell me something, I might trust it a little bit differently than if I hear somebody else. Did you say it was pick on Willie or not pick on Willie? It's what? We're back to picking on Willie. Okay, so when I hear something, I shrink it depending on how trustworthy I think it is. So if this was based on 1,000 offspring, it would be totally trustworthy. If it was based on one offspring, you could see it could have been this one here, or it could have been this one here, and I would have ended up with a completely different idea about how good the bull was. So if I only had one offspring on the bull, I'd want to shrink it towards average a lot. If I had 100 offspring on the bull or 80 offspring on the bull, I'd only shrink it a little bit. So if I had 100 offspring, I'd say his EPD was 8 to 9 pounds, even though his progeny average was about 10. And it's one of the surprising things where sometimes we see in magazines somebody complaining and saying, this EPD system isn't right, this bull's got this EPD and it can't possibly be right. Well, it kind of has to be right, because to get these EPDs, it's got to be based on deviations of a huge number of offspring. So if it's not right, then there's something else that's gone wrong. In the theory, these numbers here reflect exactly what we're seeing in the data that's coming through the system. Okay. So if this sire has an EPD of 8 to 9 pounds, which we determined from progeny testing, that tells us if I use this bull again, I expect his offspring to be on average 8 to 9 pounds heavier than those from an average sire. And some will be more and some will be less, but on average it will be 8 to 9 pounds. And I can't tell which ones are better and which ones are worse unless I purchase more information on the animal. So we buy accuracy, we pay something for it. We might pay time for it, we might pay measurement activities, or we might pay registration and evaluation fees, but we buy accuracy. So that's the kind of thing that I was taught in terms of what is an EPD. It's just about the performance of the offspring in an imaginary progeny test. But at the time I was a student, there was a lot of stuff about genes and DNA that didn't seem to have anything to do with cattle and superiority that you could see in the field. And what that theory said was that we had these chromosomes, they were in pairs, one came from the mother and one came from the father. There's 30 pairs of normal cattle, and a typical chromosome has a sequence of bases. They all have sequence of bases, A, G, T or C, adenine, guanine, thymine or cytosine, and there are 100 million of those that make up a chromosome. 30 chromosomes, so there are 3 billion base pairs. So DNA is just a chemical. We hear a lot of bad stuff about chemicals. It's just a chemical made up of 3 billion things jammed together in a particular sequence. And about 1.5% of that, representing by a few of these blue boxes here, are genes. The rest of it is kind of junk. Now those 1.5%, those genes, there's about 20,000 genes. We used to think there were more than that. I was told when I was a student that for every protein we had a gene. And we know that an individual can produce more than 1 million different proteins. So we thought there must be 1 million different genes. But as more information has been developed, the numbers kind of shrunk back. And now the number they're talking about is more like 22,000 genes. And they're recognizable because at the beginning of them, they have a kind of a header, a start sequence, and at the end of them, they have an end sequence. So somebody who is a molecular biology nerd can look at that sequence of A's and G's and T's and say, here's the beginning of a gene, and here's the end of a gene. They can count how many there are. And the way you make 1 million proteins when there's only 20,000 is some of them cooperate together on occasions, like they do in the immune system making antibodies. You can make millions of kinds of antibodies even though you've only got a small number of genes. Okay, so that DNA is a sequence of A's and T's and G's and C's, and one of the remarkable things about it that Watson and Crick discovered was it had a double helix structure. And whenever you have A, it's bound with a T, and whenever you have C, it's bound with a G. and it has one end, a 5' end, and it has another end, a 3' end. So it's kind of like a ladder, but it's twisted. It's very, very long, 100 million of these rungs in it. But if you start to rip the ladder in half, and everywhere you have an A, you join a new T on it, and everywhere you have a T, you join a new A on it, then you'd end up with 2 exact copies of it. So that means whenever a sperm and an egg come together and you have a single cell that's got all the DNA information, and each time it divides, each cell needs to have all of the information. Now, unfortunately, when you copy things, you make mistakes. And even those of you who deal a lot with numbers, if I gave you 100 ID numbers and asked you to write them out in another column, on average you'd make about 1 mistake in those 100. You'd get 2 around the wrong way or some other kind of error. Well, it turns out the same thing happens here, and an error is made about 100 every 100 bases. So that says 1 million errors are made when a chromosome is copied. And if 1 million errors were made, that's about 1% of the chromosome. You'd have a disaster very quickly. It wouldn't make sense anymore. So there's actually genes that run along and fix up these errors, and they're really good at repairing the errors. There's several genes that do this fixing. They only let a couple go through. So on average, each time a chromosome is copied, you get 1 or 2 or 3 errors that slip through. We call those mutations. And the most common one is where just one of these base pairs gets changed to another letter, and that's called a single for just one nucleotide, which is what these bases are, and a polymorphism. Poly means many and morph is type, so many types. It's a single nucleotide, many type kind of mutation. There are other ones, deletions and deletions and so on. So those little blue boxes that represented a protein, it's a sequence of A's and T's and G's and C's, and it's read in a triplet code that was identified a long time ago, and those triplets relate to something called an amino acid, another chemical. And a sequence of amino acids is called a polypeptide, or a big enough one, it's called a protein, right? So all the things you've heard about testosterone, or androgen, or estrogen, or leptin, they are just proteins. So a gene exists for every protein. This is a gene you might have heard about. There was a lot of talk in the industry about it. It's a gene called leptin. And when I was a physiology student, you know, if you asked me what is an organ in the body, you know, I'd have said, maybe not the first one that would come to mind, but later on I'd think of kidneys or heart or liver or something like that, right? and the brain and so on. But it turns out that one of the other organs in the body is actually fat. So I would have thought when I was a student that fat was just kind of a storage place when you ate more than you needed, sort of like a bank account for energy, that you put it there and you pull it back later when you needed it. But we actually now know that fat is a legitimate organ, just like liver and some of the other organs. And fat makes a hormone called leptin. That's why it is an organ. It's making hormones. And this is what leptin looks like. If you could look at the string of all those amino acids and actually visualize it, and there's machinery around to do that, and there's people, you might think we're nerds, but there's people that spend their lives looking at the shape of some of these proteins. So this is what leptin looks like, and here it's rotated 90 degrees. Those amino acids, some of them are hydrophobic, some of them are hydrophilic, they've got electrical charges, so they don't play well together. They only play well in a particular conformation. So as the DNA gets transcribed into leptin, it has this shape, and it has some bits that hang out the side, and it's got some inside bits. So leptin is made in the fat, and it cruises around the body looking for some other hormone to play with. And the hormone it plays with is called leptin receptor. So a lot of hormones, a hormone that have a receptor. So this is the shape of leptin receptor. And again, the same people that look at the hormones look at the receptors. And here is what happens when the two join together. So here is leptin, and here is leptin receptor. So it's a bit like a baseball and a baseball mitt. They fit together, right? And so when leptin joins together with the receptor, it's a switch to turn something on. So one of the things that happen when a lot of leptin is produced is it's meant to make you feel like you've eaten enough and suppress your appetite. So leptin is a hormone associated with appetite. It's also a hormone associated with fertility. And so some of you would know that if a cow is way too skinny or if a cow is way too fat, it has more difficulty getting pregnant. Why would that be? They've all still got ovaries, right? What's the difference? Well, it's things like leptin produced in this organ, the fat, that's cruising around telling the animal whether it makes sense for it to get pregnant or not. Okay, so there's leptin fitting into the leptin receptor. So what happens when we make a mutation in the gene is that we might mess up the shape of one or other of these pieces. And if we mess up the shape in the hair where they join together, it could be catastrophic because they'll not work anymore. And so in mice, for instance, if they mess with this, you get incredibly obese mice because they don't know to stop eating. But if you have a mutation somewhere else out here, it might not matter very much at all. So where a mutation occurs in a gene affects whether it's catastrophic or it only makes a minute change. Okay, so here are some pictures of leptin and the receptor. So here's the one I just showed you, cattle. And here's leptin on top and here's the receptor at the bottom. So the ball and the baseball mitt, and it's rotated two ways. Here is it in a horse, here's the rat, here's the crab-eating macaque, here's a platypus from Australia, a sheep, an African elephant, a Sumatran orangutan. Aren't they amazingly similar? If you actually look at their sequence of their DNA, they might actually vary quite a lot, but the critical piece, where the ball fits into the mitt, has to be almost exactly the same. So if you were to take leptin out of a dog and you were to put it in your bloodstream, you would respond much like you would with your own kind of leptin. Okay, so we know from this that you can't mess catastrophically with any of these hormones like leptin or they don't work anymore. But you can mess with them around the fringes where you don't change the shape of it very much. And you had a whole lot of discussion this morning about heterosis and about inbreeding and what happens when you cross animals and so on. And that was mostly based on observational studies where they crossed two breeds and looked at what happens. But now we know that one of the likely causes of heterosis is just catastrophic damage to things like this. Well, maybe not things as critical as leptin, because that would be very, very serious to mess with. But there are a lot of things that are a lot more minor that can be messed with. And Willie, sorry for picking on you, Willie, but you talked about hooves before. So I was involved in an experiment that came about when some calves were born and their hooves fell off. So within hours of birth, their hoof just fell off and exposed this horrible bloody tissue. And those calves died. There was obviously something wrong with them. So we ran a genome scan on them and we found that a protein, a collagen protein, that was responsible for holding the connective tissue was damaged in them. And it turned out that exactly the same gene occurred in humans. It's a disease called epidermolysis bullosa. And it's known in humans and it's known in cattle. It was actually exactly the same amino acid that was broken in the human form and the cow form. But in the case of the calves that we had seen, the father of the calves, it was a de novo mutation. So his gene he got from his mother and father, there was nothing wrong with those, but there was something wrong with his one. So when we mess with something critically important like this, it becomes catastrophic. But when we mess with something a little bit more minor, it often is allowed to spread a bit through the population. And so what we think happens with inbreeding depression is that you're getting two versions of a broken gene, one from your father and one from your mother, and it's completely taking out something that's not critical. And then when you cross different breeds have different broken genes. You produce an F1 that has at least one functional copy of every one of them. And that's why like Willie had said, when you cross these Angus and Simmental, you get animals with better hooves. That's the kind of explanation it could be. So all of these breeds are carrying broken genes, but when you cross them, you get an animal that doesn't have nearly as many broken genes as you do in a purebred. So we get a lot of these minor non-catastrophic things that might change the shape of a hormonal receptor and might subtly change performance, like weight. So this particular gene here, there might be a variant of it that reduces weight by 4 pounds and another variant of it that increases weight by 4 pounds. So these are not catastrophic changes, these are minor shape changes in them. And so the genetic description of why one bull has offspring different from another is that it's due to a whole of mutations throughout the genome that all sum up to give the EPD that we see. So here, if you sum up all of these numbers here, you get plus 10, and half of that is the EPD, and it's half that because you have to sum it up across both pairs of chromosomes, but a parent only passes on one or other chromosome in each pair to the offspring. So the real genetic merit of an animal is called the EBV, or breeding value. It's twice the progeny difference. And it was Richard Willem, I believe, who thought that it didn't make sense to put EBVs in sire catalogues, since people were buying a bull based on what the offspring would be like. So he said we should divide it by 2. And that's what happens in the US for dairy cattle and beef. But it tends not to happen in chickens and pigs or elsewhere in the world. You'll see EBVs, and you've got to halve them to work out the progeny value. And I deliberately showed you here one mutation that doesn't occur in a gene. So we know that the part of the genome, the 99% or 98.5% that's kind of junk, some of it actually does something, but we don't understand it. The only bits we understand are the gene parts, the 22,000 of them, and even those we don't all understand. I showed you the shape things. I think only about 600 of the genes we know the shape of the proteins they produce. So there's still a lot to be learned, and a lot to be learned about why these things here happen. But what the genetics concepts tell us is that if the EPD is 5, the true EPD is 5, then the sum of the values of all of those genes must add up to 5. That's how it happens. So if we were to have several different bulls with different EPDs, what they've got is just different variants at a particular gene. So if you look at this particular gene, I've got +5 or I've got -5. I end up with different EPDs just because each individual has got different combinations of them. And one of the things you'll notice is the EPD that's biggest here, 5, that bull still has some negative variance. And an EPD of -3, the worst bull here, has some +1s. And that's why an average bull can produce an occasional amazing offspring, and amazing bulls can produce some very poor offspring. Okay, so SNPs. Those SNPs I talked about, they occur in junk areas. They're not messing with a protein, it doesn't really matter. So SNPs in junk areas accumulate. So the junk areas of the genome are full of these mutations, whereas the important things of the genome, like leptin, are not that full of SNPs. I showed you before how similar they were even in those diverse species. So this was from one of the first chips that came along here, and if I'd known Mike was in the audience, I guess I would have done an Illumina one. But this was the first chip long before the Illumina 50K. This was a 10K chip that came out, and they tried to put SNPs on it that were all across the genome, and you can see there are a few places with big gaps, and there are other places where they've got too many, but they've got them across all 29 of the autosomes. They didn't have them on the sex chromosomes. What's revolutionised it from our point of view is these Illumina products, and here I show you 3 of them. And they're little things. Bruce actually had one in his bag. He might hold up if anybody wanted to pass it around so you can have a look at it. But this was something that Mike had distributed in, at the BIF meeting, so anybody can open that up and pass it around. And I'm not quite sure how many, there are 12 lanes on that maybe. So each of these lanes is where the sample from one animal would be analysed. And if you were to look inside that chip, what you'd find is there's been little holes bored, and those holes are 2 microns. Now I know out west here there are a few sheep, and if you were to pull the fibre, one woolen fibre, and hold it up, it's probably, in the sheep out here, probably something like 30 microns. That'd be about right for those sheep people here. A really, really fine merino would be under 20 microns, maybe 15, maybe even 14 or 13. So these are 2 microns, so they're minute holes, but they're made to exactly fit a 2 micron bead. And if these 2 micron beads are dropped on there and fall in the holes, they get kind of sucked in so they don't fall off. So once they're in there, you can shape that chip and they won't fall off. And if you look at every one of those beads, they've got a little piece of sequence hanging them off. And one of those beads of 2 microns has about 800,000 of those little whiskers hanging out. That's got a sequence that corresponds to a particular piece of the genome. And so what happens is if you send your DNA sample or your hair sample, hair doesn't have DNA, but the follicle, the bottom that extrudes the hair does, then it gets amplified and it then joins up to where the bead has the corresponding or the opposite piece of sequence, so it finds its complement, and they refer to that as hybridization. And then it gets labeled with fluorescent dye. And if you were looking at Bruce's pictures, maybe some of them you would have thought totally reasonable. I don't know if you recognise the one of him sitting next to one of those old computers, but there was one there in his earlier days. But maybe you didn't recognise those ones that were red and yellow dots, right? The red and yellow dots was the picture of one of these chips when it's lighted up with fluorescence, because those beads either turn green or they turn red or they turn green and red according to the sequence at a particular snip. So when they shine a light on that little chip, in any one of those lanes there are a huge number of beads, and each bead lights up, and according to the light on it they can tell what the particular mutation is that a particular animal carries in a particular place. So that's how it works. So that was the picture he showed. That's what's read by the microscope that reads these chips, and based on that they will tell you whether you'll say CC, GA or TT at that particular mutation. And each of them are named here like I've shown you, and that comes back to people like me as a genotype report, and they just tell me an animal is AA or AB or BB. So if we were to genotype these 3 bulls, this is what I would be told. This bull is AB, this bull is BB, and this bull is AA. And that would just respond to the 3 different colours that the chip fluoresced after the analysis had been done. And so this is what you are buying. This is the information you're buying when you send your hair sample off to Sovietics, where you send your hair sample off to GeneSeq. You're just buying an AA, BB or an AB genotype. So now I need to tell you about what happens, because those SNPs don't get inherited independently. They get inherited in blocks. So I know, for instance, that in my case I have about 5 million SNPs that make my genome different from the reference human genome. But those 5 million don't get inherited one at a time. They get inherited on one of the 23 pairs of chromosomes that we have. So if I show here one pair of chromosomes, and I'm showing one blue and one red, representing whether it came from your father or your mother, if you were then to collect a gamete from this bull, a sperm sample from this bull, or an egg from this cow, and you were to look at that one chromosome, what you'd find about 30% of the time, that gamete would carry exactly one or other copies of the chromosome the individual got from its father or mother. More commonly, like 40% of the time, there would be one crossover event, and that means these 2 chromosomes got tangled up when they were being copied and produced a gamete that is partly from one parent's chromosome and the rest of it is from the other parent's chromosome. And that recombination point occurs anywhere from one end of the chromosome to the other, but it does occur more often in some places we call hot spots and less often in other places we call cold spots. And then occasionally, like 20% of the time, we get 2 crossovers, and 10% of the time we get about 3 crossovers, and we virtually never get more than that happening, at least under normal circumstances. So if you were to look at all of the sperm samples that came from a bull, you'd find, say, some were all blue or some were all red, representing they came from the bull's father or the bull's mother, but a lot of them would be mixtures, and some of them would be mixtures with more than one colour. And we've actually looked at how many times a crossover occurs in offspring of one bull using chip data, and it actually varies from one bull to another. It's a heritable trait. So if you wanted to add to your list, Bruce, you would add the number of crossovers varies from one bull to another. So there are actually genes that control how many times a crossover occurs. Okay, and there are major genes affecting that. So if we were to look at just one little piece, maybe sitting over a gene, let's say like leptin, a little piece, say 1% of the chromosome, so it's only 1 million base pairs, then we could separate half of the sperm to being ones that got the blue bit there and half of them to being ones that got the red bit there. Okay? And if we could look at the value of that fragment, we might find the value of this fragment is -4 and the value of this fragment here is +4. So that little fragment might contain 10 or 20 genes in it. And I might not know exactly which one it is, or even if it is a gene or a junk that's causing a variation in birth weight or weaning weight or any of the other economically relevant or indicator traits you're interested in. But I can tell that there's a difference in value between the offspring that got the blue piece there and the offspring that got the red piece there. And that's what happens when we do a genomic analysis. And we work out the value of those pieces by doing regression. So we just regress the breeding value or the progeny test value against how many copies of the blue alleles or the red alleles, and we get the slope of that line. But we use multiple regression, so we do it for all the genome all at once, so that involves a bit of computer analysis and some of the equations that Bruce had for you before. So if we come back to that original bull, I said he's got a gene here that maybe has a variant that reduces or increases the weight trait. I don't know that. I don't know the mutations there because I've only got one over here from the SNP chip. But I can recognize that piece as being blue or red, and I can work out the value of it, but I have no idea of which of the 1 million base pairs in there is causing that difference in performance. But I can say the blue bit reduces weight and the red bit increases weight. There's 1 million base pairs and I don't know which one in there precisely it is, but I can see this based on what's happened on a bull we've seen before. So when we get a genomic EPD, what I'm doing is I'm just breaking the genome into fragments using these SNPs, and I'm getting an EPD for a fragment rather than an EPD for the whole bull. And if I sum up the values of all of those and divide them by 2, that's what the EPD is. So the genomic idea of an EPD is we look at the genes and we work out the values of the genes. But if we don't know the genes, we just look up the fragments that we can follow and we work out the value of the fragments and sum them up. If I didn't know about genomics, I'd work out the value of all those fragments by doing a progeny test. But either way, the underlying mechanism is exactly the same. It's exactly the same thing that causes a bull to be good and bad as these genes. So when you select on a progeny test, you're visualizing this stuff here very, very crudely because all you're seeing is this number here, which is kind of like an index that's summarizing all of the genes that are in there. But if I'm using genomics, I can look at each particular position. And at each particular position, I've got a number of different haplotypes, all these colors that I've shown you. So does this work? So we can do this, no problem, we can do regression. One of the things you might know about regression is it's incredibly accurate telling where you've been when you look in the rear vision mirror. But what we we really want to know is this any good for telling where we're going to be next? So what we do is we take the population and we split it into bits. So we might split it into 3 pieces. And we do we characterize whether it's a blue or a red fragment. We work out the value of it in 2/3 of the data. And I apply that to predict the individuals in the group 1, which I wasn't using in my regression analysis. So in order to do this, I have to group the animals into groups. And this used to be easy. I'd take a couple of thousand Simmental Association animals, and we'd split them based on pedigree into a few groups of 700, and we would have no trouble. We could do this cross-validation. But now we're up to over 30,000 animals that we have genotypes on that have offspring in the Simmental Association database. So if I go and try and cluster them, I find out that I don't get like 10 nice groups of 3,000. I get one group that's got 27,000 in it, and I get a hangers-on. So we have to come up with a nice way of grouping them. So we've found out that it's better to try and group them based on the markers rather than basing them on the pedigree. So for anybody who's done statistics, this is a 3D plot that's looked at in 2 dimensions that looks at the first 3 principal components of the genotypes. So to explain that in rancher language, if I put a whole lot of cattle in a pen and asked you to sort them out just on whatever you thought, you would look at them, and if there were big ones and little ones, you'd probably put the little ones together and the big ones together, and if some were fat and some were skinny, you'd put the fat ones. And so you would look at that group, and you would something would poke you in the eye as being different about some animals and other, like their height or their colour or their fatness. So these principal components are the thing that's poking the computer in the eye about what's different about the genotypes. And I'm showing it to you in 3 dimensions, and we colour them according to the breed. So these ones here are Black Angus bulls. So these are pure Black Angus bulls who have progeny in the Simmental Association database. And what you'll notice maybe is there's a cluster of them here. So the computer says these guys all look pretty much the same. But there's a little hidden one here. There's actually another cluster of Angus here. So the computer says when it looks at 50,000 genotypes, it actually sees 2 clusters of Black Angus bulls. And I don't know what it is that's making one of those clusters of Black Angus different from the other. But it also sees clusters of Red Angus. And what's interesting is the Red Angus it sees as being very different from the Black Angus. They hardly overlap at all. And there's one cluster of them here, and there's another cluster of them here. So there's also 2 clusters of Red Angus. And then there's some Mainonju, the RDPs here. There's one cluster of those, although it's kind of a stretched out cluster. It's not a tight cluster like the Black or Red Angus. And then there's Geltbees here, and you'll see there's actually 3 strung out groups of Geltbee, sort of stretched in these directions. And some of them are looking closer to Angus, and some of them are further away. And then all of the other spots, purple is it, are Simmental. So Simmental is a much more diverse. So you were talking about, earlier this morning about whether you were an industry thing or a breed thing. If we did this in Europe with Simmental, they would be a tight little cluster like we're seeing for Angus or Red Angus, right? They wouldn't see a cluster looking like this. So the immediate thing you think is, well, what's different about those Simmental? So I had my postdoc who was looking at these to have a look at the breed fractions. And so the black dots are ones that are full bloods. So you see in here there's a bunch of black dots here, but there's also a bunch of black dots down here. There's also a bunch of black dots down here and a bunch of black dots down there. So the full bloods actually cluster in 4 groups. And don't ask me what's different about those 4 groups. I can give you the IDs and you can tell me what's different about them. And then there's a bunch that are recorded in the database as being more than 95%. And when we colour those with those light blues, you see they sit almost on top of where the black ones are. So those 95% and above, we can't distinguish from a full blood in terms of looking at their genomes. And then the next ones down would be the 87 to 95%, and those are the ones that are blue. Again, there seems to be 3 groups of them, and they are moved up a bit. They're not sitting right on top of 100%. And then there's ones here in purple that are 60 to 80%, and there's the ones that are 60%. And you notice the 60% ones are kind of sitting in between the full bloods and the Angus, because they're probably some Angus cross. So what we're doing now is we're taking this diverse population and we're clustering them, and then I do the regression on everybody else. So I work out the value of all their genome fragments, and then I try and predict these ones here, having left them out of the analysis to get a validation of how well things are working. So I don't have the results to tell you how those are working from the 30,000 animals that we're running that evaluation now. But I do have the results based on the prediction equation you're currently using. And I just explained how we could do it by partitioning in 3. So if we partition in 3, we don't like ever throwing data away. So we would do another analysis where we leave out group 2 and validate group 2, and another analysis where we validate in group 3. So when we're finished, we validate on every one of the individuals. And what I show you here are the results of this cross-validation for a bunch of traits, for a bunch of breed associations. So these predictions are based on prediction equations that are currently used in these breed associations, with the exception of the Angus one. So the Angus one is one we publish, but it's not used by the Angus Association. The Angus Association use a Zoetis prediction. But there's a red Angus one, which includes red and black bulls. There's a Hereford one, the Simmental, the Lindsay, and the Gilpie. So these are based on the current predictions that are available at GeneSeq. We have a lot more data than this for all of these breeds, and we're just in the process of updating them. But on average, the correlation, which is the strength of the association between how good I think they are based on summing up the values of all those fragments, and how good they really are based on being progeny tested. So we would like that number there to be a number like, 0.8 or 0.9, but on average here it's about 0.5. Some of these traits it's in the 0.6s, and some of them it gets up a bit closer here to 0.7s or maybe even 0.8s. But generally speaking, as we get more data, those numbers get higher, and we expect them to get higher still as we go up from the current data sets to the ones from what we had here in the predictions to the current data. So Hereford we have over 10,000 now. Simmental we have over 30,000, partly because you use all those other breeds in your association, and partly because you've been a more aggressive adopter of this technology, but largely because the Canadians have been really good at leveraging public money to subsidise their efforts, and it's been a huge boost to us getting data from all of these breed associations from Canadian efforts. So how all this works, the National Bead Cattle Evaluation Consortium and postdocs, we have at Iowa State develop prediction equations, we put them at GeneSeq, you send in your hair sample, GeneSeq runs a bit of software we've got there that generates the predictions, and then the breed association can pull down the predictions and the genotypes. They do a blending process and give them back to you. So sometimes I get a phone call or an email asking me something about a particular bull, and I have no idea what you're talking about, because although I have all the historical data and develop a prediction equation, unless I go to the GeneSeq database and pull down the genotypes, it's not easy for me to look and see what's happened and find out what's going on. But strangely enough, I've never had a phone call asking me why an animal was better than they thought it was. I quite often get them where they think it's a whole lot worse than they thought it was. So right now a blending process is used, which is not what we would like to see happen. We'd like to see an analysis done together. But this is a graph showing you the BIF accuracy without any information, and without any genome information, and BIF accuracy with genome information represented here by the blue line. So the black line is just Y/X, nothing to change. The blue line shows you how much increase you get from having genomic information. And it depends on how accurate the prediction is. And that varies from trait to trait and from breed association to breed association, depending on how much data they've got. So if the genetic correlation is 0.7, this shows the lift you get. So you get a pretty good lift if the VIF accuracy of the animals you were testing is 0. But if the VIF accuracy is above 0.6, you get virtually no information from the test. So one of the things that people who haven't thought about it might think is that genomics would help you when you do it to a proven bull, but of course it's not going to change anything because you already have an accurate EPD. Okay? So that does raise the question of why would you genotype an accurate bull? And you wouldn't genotype it to help you know more about the bull. You genotype it to help everybody else in your allegiance here get better predictions the next generation round, because we need to know the values of those accurate bulls to work out the EPDs of all the chromosome fragments. So when you genotype an old bull, you're investing in better predictions in the future. You're not learning anything really new about that new bull unless you look precisely at some of the genes it has. If you have a higher correlation, a genetic correlation of 0.8, which we're getting up to that value in some of the traits and some of the breeds, now we get a much bigger lift. And now the return extends a little bit higher. It tends up to maybe above 0.7 compared to having pedigree only. So you get a bigger return on genotyping investment when you have a higher genetic correlation. So my goal is to try and get this correlation as high as possible. And we're using all kinds of methods to do that. One is by actually looking at sequence, to looking at the actual genes, it's looking at what's happening in other breeds and other species. It's pulling together data from other breeds and seeing if we can get that to help us identify where these things are. So the other thing that's important is that some people think when you buy a genomic EPD, you're buying a better EPD. But that's not the case. What you're buying is accuracy, and it's equally likely that the EPD will go up or go down. That's a feature of the EPD system. The B in the block that Bruce told you about stands for best, and the U stands for unbiased. So the predictions are best and they're unbiased, and a consequence of that is you can't do better than the information we give you in the EPDs, which is why using the indicators as well as the ERTs doesn't help. And they're unbiased, which means that we're equally likely to have underestimated or overestimated a bull, which means whenever you buy more information from progeny tests or from individual performance testing or from genomics, they should be equally likely to go up or down. So one of the other fascinating things about all of this is that when we look at those little colours, we can find out where on the genome the big things are occurring. And here I've numbered the chromosomes. They're numbered, strangely enough, from 1 to 30. And here on chromosome 7, at about between 93 and 94 million bases along the chromosome, and you can go to a database on the web and you can look up that chromosome, that position, and you can see what genes are there. There's a little fragment that in Angus accounts for 7% of the variation in birth weight, and Hereford accounts for about 6% of the variation. Doesn't do anything in short-horn limosome, not much in Simmental or Bead. There's one on chromosome 6, and it's right at the end of the 38 millionth base, somewhere between 38 and 39 million boundary. It only had a small effect, about 0.5% of the variation in birth weight in Angus, but it was quite big in Hereford, almost 9%. It was huge in Shorthorn, 12%, pretty big in Limousin, 6%, but it was 16% in Simmental. So that one little gene fragment, there's something going on there that is having a huge effect on birth weight in Simmental. And we also see that same fragment in Guelphy and Limousin and all these other breeds. And we looked at another method of doing the analysis in Angus, and the amount of variation in this region went up a lot. And that really just told us that the 50K chip we were using is not very good in Angus, in that particular little fragment of the genome. There's another one on chromosome 20, about 4 million bases, that has a big effect across a bunch of breeds. And there's another one on chromosome 14, and this is an interesting one because we actually know the causal mutation in this gene, at least we think we do. It's at 25 million bases, and it's in a kind of an unusual part of the genome. It's part of the genome that's incredibly gene rich. And it's incredibly gene rich, it means it's got lots more GCs than it's got A's and T's. And that means it's really hard to sequence and it's really hard to genotype. So the 50K chip is actually really bad in that particular region of the genome. But this particular window, we call it a QTL, or quantitative trait locus, was published a few years ago as being an incredibly big gene in Wagyu cattle by a Japanese researcher. And I had been involved in New Zealand in designing an experiment to cross Holsteins and Jerseys and try and find genes that made Holsteins different from Jerseys. And in that experiment, we found a really big gene in the region somewhere around here that was responsible for a lot of the difference in size between a Holstein and a Jersey. But it was about 1 million bases away from the one that the people in the Wagyu thought was the causal mutation. So some of my colleagues went on and they found what they believe was the causal mutation, and it's a gene called PLAG1, PLAG1. And they got that mutation, and Holsteins are almost all homozygous for the big form, and Jerseys are almost all homozygous for the small form. And what really surprised me when we ran our analyses, and this popped up as quite a big gene effect here in Simmental, and an even bigger one here in Guelphy. And then it surprised me even more when paper came out in the law cattle, so the Indicus eared cattle that are in Brazil that came from India. This is the biggest gene affecting some of the growth traits in the law cattle. We also see it as a big effect in Brangus cattle. And then I was in Korea, and it turned out to be one of the biggest effects in Hanu cattle. So if you've been to a Korean barbecue and you have that beef, they're very, very fatty beef. It's like the Korean version of Wagyu. It's also the biggest gene there. So it's amazing that we're getting these same gene effects occurring across a whole bunch of these different breeds. This will make it a lot easier for us to find the actual gene causing these using the Simmental association database. We have all of these mixtures of breeds. But what was really interesting about this particular gene is that we knew it had a huge impact on weight, but the Australian data, the CRC, the Cooperative Research Centre, where they used government money and industry money together, they did a whole lot of experiments on gene discovery, and they collected a lot of reproductive data. So they scanned the animals and worked out when they reached puberty and so on. And they published this effect having some interesting effects on growth and reproduction. So they actually interrogated the actual gene, not just the markers like we're doing. And they said they found the difference of one copy of this being 5 pounds of birth weight, and between the ultimate homozygotes. In the Simmental data, we find the difference to be about 7 pounds. 10 pounds of weaning weight, 16 pounds on feedlot, 24 pounds off, 14 pounds of carcass weight. So we knew all that because we saw all that on your data. But one of the things we don't have a lot of data on is reproduction, because the total herd reporting hasn't gone nearly as long as the collection of births we've been dealing with. But this is the Australian data. The effect of one copy, age at corpus luteum, first corpus luteum, so this was their measure of reproduction, 38 days. So that's 76 days in age of puberty between an animal that has homozygous one version of it, between animals that have homozygous the other version of it. So this is just like, a shape change in that lectin thing that makes them stick together better and turn the switch on a bit stronger or weaker. That same gene effect, postpartum anestrus interval, extended at 15 days. So 2 copies, that's 30 days different in postpartum anestrus. Imagine what that's doing to reproductive performance. If a cow can get to be cycling 30 days earlier. So with the same conception rate, the pregnancy rate, the stability is going to be a heap better. Presence of a CL before weaning, there was a 5% reduction. Weight at first CL, 36 pounds. Age at stroke circumference, which some of you seem to be interested in, 90 days. So this gene seems to affect growth. And what do you want? You want growth, you want these bigger weaning weights, bigger feedlot weights, bigger carcass weights. But what's it doing? It's extending puberty. It's making postpartum anestrus. So what version of the gene do you think Angus has got? Remember, if we come back here, Angus, we didn't really get any effect of this gene in Angus. We didn't really get the effect of Hereford. So we've got to expect here. So what version of the gene do you think they've got? They've got this one. They've got the big growth, poor fertility one. They're virtually all fixed for it. And so their selection for the last few decades, where they've looked at size and things, has inadvertently selected for this reduced reproductive performance. It's interesting, we recently ran some analysis on some Herefords from Uruguay, and we actually get variation in that fragment in Uruguay. So probably the traditional Scottish Angus and Hereford, which were a lot smaller, they probably are segregating that plag gene just like the Hanu and the Wagyu and the other things. What about the Indicus cattle? So it turns out that the Indicus cattle, the version of this gene, is actually not an Indicus fragment. We can actually tell that that fragment came about when they first introduced the breed into South America and into Australia and Brahman into the US. There weren't a lot of them. So they crossed them with things like Angus and Hereford, and they carried through the other version of it, and it's since been selected up. So there's actually a piece of about 2 million bases in the Law cattle that's not Bos Indicus, it's Bos Taurus. It came relatively recently. So while the stuff we were doing here, we didn't expect at the outset that this genomic prediction might have an effect on reproduction. But it looks like we are going to find things in reproduction that are going to help you get better predictions for some of these other traits, even though our analysis is based on more basic genomic stuff. So summary, genomic prediction is just like pedigree-based prediction. It's based on concepts that were established decades ago. So right when the original idea of what might explain differences in EPDs between progeny tested bulls were due to summing up the effects of the genes, but nobody knew how to see the effects of genes or sum them up. So that method just sat in textbooks for 50 years until Illumina came out with a cheap way of doing all the SNP genotyping right through. Genomic prediction is an immature technology, so it took you whatever it was, we said 60 years for you to develop indexes to where you are now, but it's only taken us 4 or 5 years to develop genomic prediction to where it is today. Imagine what it's going to be like in another 50 years. Existing evaluation systems need considerable research and develop to implement all these changes that are required in this genomic prediction. So I don't know how we're doing for time, but we were late starting, and so we're good. So I'd be willing to attempt to answer or deflect any questions that anybody might have on anything to do with EPDs or the genomic things we're doing. Sally.
How about Red Angus compared with Angus on this gene that has to do with their
Red Angus, I'm just not sure off the top of my head whether we saw that one segregating in Red Angus or not. One of the interesting things we see, I showed you a little example where I looked at 1% of the genome and I said an offspring of one bull, there's say a blue form and there's a red form. If we look at another bull in the same breed, there might be different haplotypes or different colours. And on average, in a little 1 megabase region in a breed, we see about 18 different colours on average. And one of the reasons we can't do a crossbreed prediction is that the 18 colours that we see, say, in Charolais are completely different to the 18 colours we see in Hereford, and completely different to the 18 colours we see in Angus. If we compare the colours we see in red Angus to the colours we see in Angus, we see that all the colours we see in Angus exist in red Angus, which is no surprise because black Angus bulls are allowed to be used in red Angus, but we don't see it the other way around. So we see a whole lot of colours in red Angus that we don't see in black Angus. And what that means is if we do genomic prediction in red Angus, it actually works in black Angus. But if we do genomic prediction of black Angus, the prediction equation does not work in red Angus. So I was a little bit surprised when I saw those graphs to see how red Angus and black Angus are sitting a little bit apart. But I think that's probably reflecting the differences in selection policies over the last few decades. So it wouldn't be surprising because of their emphasis on reproduction to see that black gene may still be segregating into red Angus. But the data from Clay Centre, if I remember rightly, and maybe Matt knows, I don't think there's any real difference that they see in the mature size of red Angus and Angus in their germplasm variation. Red Angus is a bit smaller? Okay, so if red Angus is smaller, then there's a good chance it is still segregating that more fertile black gene. But if you are interested, as Wade said this morning, in doing something that's good for the industry, I would have thought what you would want for the industry, you would want mother cows that have that version of the gene that make them more fertile, earlier puberty, shorter rebreeding interval, smaller size. It's going to help you on income, it's going to help you on costs, and then you want to make a bull to them that's got the high growth version of it, right? And that would be like crossbreeding, and you could do it even though the animals might all be the same breed. And so what we're trying to do now is predict the effects of those particular genes for particular bulls, so we can show you which version a particular bull carries. And we think if we could characterise just those 4 that I showed you there, we could get pretty good predictions for a whole range of traits, birth weight, calving, easing, and so on, just from the effects of those few genes. And that's why we started sequencing quite a few animals. We've sequenced 50 Angus sires, and if I had my choice I wouldn't have done Angus sires, but we only have an Angus herd, so I've sequenced 50 of the Angus sires that we have used in our 400 cow Angus herd in Iowa. So we have sires and grandsires of most of the cows we have now completely sequenced. And we have put that data into an international consortium known as the 1,000 Bull Consortium, which interestingly has 1,147 bulls in it, and it goes up all the time, but it has them from a whole range of breeds, and it has a huge number of Simmental in there from Europe. So by putting in my 50, I get access to the 1,147. So we're trying to use that data to try and help find the actual mutation. But the flag one, which we believe is the correct one, unfortunately does not work on a snip chip. So these snip chips are magic, but there's some things they don't do. And one of the things they don't do very well is do genotypes in these GC-rich regions. It happens in the Holsteins in Jersey that there is a snip that works very well at picking up the effect of that plague variant, but we haven't found one yet in the beef breeds. So we're trying to do more work in that area to try and come up with a snip that will do a good job of predicting the presence of that plague one, because I think that plague one alone could be hugely valuable to people like you who have a vision. But you're going to have to change your way of thinking about this. You're going to have maternal bulls that you select particularly to have that gene that affects positively reproduction and smaller size, and then marry it up with bulls that are the other way around. And you can't do that if you have this vision of one size fits all, right? Because you're going to have conflicts.
So could a Simmental and an Angus both have that or?
So it looks, I mean, the way it looks at the moment is that the Angus are all fixed for the big size, late maturing version, but the Simmental, some Simmental are still carrying the smaller, more fertile version of it. Now what I don't know is I can't tell you the name of the bulls that have that. It might turn out that they were ones you stopped using 20 years ago because they were too small. Or maybe it's a model, we don't know that yet. Well, we do know that among the animals, the Simmental breed animals, Simmental breed association animals that we have genotyped, some of them carry one form and some of them carry the other.
And none of the Angus.
Virtually none of the modern American Angus do, but I'll bet you anything you like, say a lowline Angus is probably homozygous for that other form. That's one of the things that made them get so small so quickly. Yes, we do. We could look at the SNPs and track down where these things. So one of the things about these genotypes is we can look back a long way, and you may have seen some things in the news, recently about Neanderthals. So there's only 8% that have more Neanderthal than me, which may or may not come as a surprise. So there are no secrets when you want to look inside. You can tell a lot about history by tracking these fragments. So yeah, we will be able to track them a lot more, but higher density allows us to track them more accurately than lower density does. And sequencing helps, at least in some of the regions. Sequencing is not the overall secret, because only some parts of the genome sequence easily. Some parts don't sequence very well at all. So sequencing, you learn a lot more about some parts of the genome. You miss out on some parts, at least with current technology.
Going back to that, when you were talking about the lore, I guess you were still talking about this PAC1 gene.
PAC1 gene.
And it doesn't have a smooth.
It doesn't have a ZB background, no.
But the lore don't mature sexually very early.
Right. Right.
So.
Yep. Yes. So the version that they have inherited from the Taurus is the later maturing version, and the reason for that might be because they're in a tough environment. It's an advantage for them not to get pregnant as early. If they get pregnant too early and they're in poor condition, they don't do very well, they end up being culled. And so artificial selection might have been favouring the ones that got pregnant later and didn't get as pregnant as easy because they did better in the tough environment.
Well, they also might be the same thing when conditions are really tough and when cattle get thin. Prominent and lower cattle don't reproduce.
Right. That's right. And I mean, no doubt there'll be a whole bunch of other genes that are involved in fertility, but that's just one of the interesting ones that's popped out recently.
So how do you know it's a Taurus instead of
Because the haplotype that is in it is exactly the same haplotype that's in Angus and in Hereford. It's a taurine haplotype, and the haplotype that's associated with the other level of performance is completely different, and it's one you don't see in Taurus. So there's a paper that came out about, I think the end of last year, by Marina Fortes, Fortes, that has a lot of interesting information. You should read that, Marina Fortes. If you email me, I can send you the exact reference, but it came out in 2013 on the PLAG1, validation of the PLAG1 gene. And they validated a whole range of breeds, but they didn't have much data. They might have had a couple of thousand animals, but it was over about 20 breeds. But they did have really good reproductive data because they had scanned repeatedly those heifers right the way through to see exactly when they reach puberty. Australian Angus are fixed for that big form, which is no surprise because Australian Angus are just, a huge impact from US Angus that went into Australia through New Zealand.
So we have both growth and a phenotype
there was some talk earlier, I might be interested about not much genetic diversity, and at least what we see in the genome, that's not the case. So we have a term we refer to as the effective population size, and So the amount of inbreeding in a population is related to the effective population size. And you might have heard this term bandied about. So the rate of inbreeding in say, Holsteins is equivalent to what you would have got if you had about 100 individuals breeding with each other. Okay, so it's an effective population size of like 100. And Hereford, it's like 100 or 200. On Green Angus, it might be 300. It depends a bit on how people estimate. But generally in the beef breeds, it's sort of in the 100 to 300 numbers. In humans, the number's more like 10,000. So the effective population size of humans is 10,000. So we're a lot less inbred. So we would expect to have a lot less diversity in cattle than we do in humans. But we actually see more diversity in humans in cattle than we do in humans, or the same amount. And the reason for that is that the diversity in cattle, the effective population size has shrunk since domestication very, very recently. So cattle have gone through a bottleneck, so they don't have very many haplotypes. I said on average about 18, but within each of those haplotypes, they've got incredible diversity. So cattle have got small diversity in big chunks, but within the chunks, they've got the diversity that humans have. So we can mine that diversity over the next little while very easily, because is all still sitting here. It's just a matter of identifying. The trouble is, if the variant you want is one you've inadvertently been selecting against for the last 20 or 30 years, there may not be very many animals there that have got that variant. So you may have to introgress it by back crossing to some older animal, and that's why it's good that places like USDA have been storing germplasm so we can go back, and maybe we'll go to another breed and get it. And yesterday there was some talk about hairy bulls. I think, what did Marty say? He wanted slick bulls. So there's a lot of information about the gene that causes slickness. And there's a slick gene that comes from Simmental, for instance. So you could extract that and produce a slick version if you wanted to. Or there's a new technology around now, which when Marty was talking about things that were way out there yesterday, gene editing was one that occurred to me. And gene editing is a method where you can actually go and change one base pair in an animal without changing anything else. And it's being done right now. USDA is trying to produce a whole Holstein, and I believe there's some work to produce a slick-haired Holstein. And there's been a whole lot of other work done to produce pigs, where they've gone and modified one gene to make it have a defect the same as occurs in humans, so they can create pigs that have exactly the same genetic disease as occurs in humans, so then allows them to study how they can minimise the effect of that. So that's another option. If you don't have the variant that we know you want from some other breed, you can actually gene edit the zone. And that's just a way, it's a bit like transgenic. A transgenic is normally you're putting a gene in across from another species, whereas this would really be cisgenic. You're putting it in with the same species. You're not actually putting anything in or taking anything out. And the reason I think you talk about a 20-year time frame last night, the wee reason I think this is going to happen, among others, is that it's not detectable. And so you can't go up and say, I'm going to put a speed limit on this road, unless the police can go out there with a radar and tell you how fast you're going. You can't enforce a law that you can't test for. And that's the thing about this gene editing. There's absolutely nothing in there that is detectable. So you can't tell if it was a natural mutation or if it was an unnatural mutation. So if you can't detect it, then you can't actually legislate, because in court they can't show that these animals were modified. So my guess is within the next 20 years, you'll be able to go and modify an animal. So you could have gone and taken a full blood Simmental and you could have made it black by editing one base. And nothing else would have changed.
Well, for right now, can we just crossbreed a high growth Angus, a guard rabbit, with high internal Simmental, say sheer force, and get both of those?
You can crossbreed. So the problem with crossbreeding is you're going to end up with a whole lot of fragments that come from one breed and a whole lot of fragments that come from the other breed. And that might be really good to have at your commercial level, but that may not be what you want at your nucleus or elite kind of level. And I think maybe Marty's going to be talking later about pigs, but what happens in pigs or chickens is that typically they have about 10 breeding programs. So if you take chickens, for example, there are small chickens and there are big chickens. And some of the chickens, when it gets hot, they don't eat. So if you're in a hot place, you need a different kind of chicken than if you're in a cold place. And some of them get sick easily and some of them don't. So the companies have, say, 10 lines, but which 4 lines they put into a chicken in Nebraska will be different to the 4 lines that they use to put into a chicken in the UK, or the 4 lines they use to put in a chicken in a place like New Zealand. So with just a small number of lines, like 10 lines, with maybe only a couple of thousand birds per line, they can generate appropriate material for any environment around the world in terms of disease, temperature and size and everything else. So the last thing they would want is their elite population to be mixed up with all one kind. They want the commercial ones to be mixed up and not the elite ones. Mm-hmm. So it comes to something that went through my mind yesterday when you were talking about the alliance, and I didn't hear it, I think, until Matt brought it up, and that's the issue that amongst the breeders, there are what we call the nucleus breeders who are driving improvement, and there's the multipliers who just multiply it and market it. And one of the decisions a breeder needs to make is, do they want to be a driving the nucleus kind of breeder, or do they want to be a multiplier breeder? And there's a huge distinction in what you do and the way you do it, depending on whether you're trying to be a nucleus or whether you're trying to be a multiplier. And one of the things that struck me last night was from some of the discussion that came from the floor, was some people were indicating, I really want to be the nucleus, I want to be driving the change, I want to be creating the bulbs that are making things different. And then I had other people in the audience saying, hey, I really want to be a multiplier, just give me the seed and let me multiply them up and sell them. But from the point of view of the association and the services it offers, the service and things you need to be doing to be driving change are a bit different from the services and things you need to be doing a good job of multiplication. And again, it's rare to have somebody that's good at both of those things in the same operation.
Okay, let's thank our speakers. Okay, next up we've got Dr. Golden back.